Hello, my name is Nicolette Beatenbow, and this is my biochemistry final review. Our first question asks to draw an approximate Lineweaver-Burke plot for each of the inhibitor types in question 25. Question 25 was asking about no inhibitor, non-competitive inhibitor, competitive inhibitor, and mixed inhibitor. This was covered in topic 31 and in chapter 8.4. It is asking to draw a Lineweaver-Burke plot, which is another name for a double reciprocal plot. A key term is double, meaning there will be two reciprocals, one being of velocity and the second of substrate concentration. Here's an example of a double reciprocal plot versus the direct plot, and both of these can be derived from the equations down here. Vmax is going to be represented on the y-axis, while Km is on the x-axis. So A has no inhibitor, and you can see the double reciprocal plot right here in purple. This is going to be used to compare the other type of inhibitors. In B, a non-competitive inhibitor is competing for the enzyme non-competitively by binding the enzyme in an active site other than where the substrate binds. This causes a conformational change, which prevents the substrate from binding its own active site. You can see the double reciprocal plotted right here in purple, and you can see that Vmax has decreased while Km has remained constant. In C, a competitive inhibitor is binding the same active site as the substrate. You can see that plot right here in purple. Vmax has remained constant while Km has increased. In D, it is a combination of competitive and uncompetitive inhibition, so there's not a direct scheme here. The double reciprocal plot is going to be somewhere between the two previously stated. You can see it here in purple. Vmax has decreased while Km has increased. Now to the next question. Many isolated enzymes, if incubated at 378 degrees Celsius, will be denatured. However, if the enzymes are incubated at 378 degrees Celsius in the presence of a substrate, the enzymes are catalytically active. Explain the apparent paradox. This was covered in chapter 8.3, topic 37, and chapter 9. It is asking about the catalytic activity, and some key terms that came to my mind when reading this question and thinking about catalytic activity were transition state, stabilization, and denaturation. This first image is showing how an enzyme is denatured in the presence of heat. You can see that the substrate has a complementary fit for the enzyme active site. After heat is applied to the enzyme, a conformational change happens that prevents the substrate from binding. Every enzyme has an optimal temperature, which is shown right here. As you can see, as you slowly approach the optimal temperature, the enzyme becomes more catalytically efficient. As soon as you get past this optimal temperature, the enzyme catalytic activity rapidly decreases. Another thing that came to mind when reading this question was induced fit, when the enzyme binds the substrate and weak intramolecular forces form and stabilize the 3D structure. Another thing that came to mind was the oxyanion hole, which stabilizes the negative charge right here on this serine, forming this intermediate, which has more stability due to this hydrogen bond. The enzyme is stabilized by substrate binding and intermediate formation. Here is another image of the oxyanion hole stabilizing the negative charge on the serine residue. This question is very similar to another question in the Chapter 9 workbook problems asking, many enzymes can be protonated against thermal denaturation during purification produced by the addition of substrate. Propose an explanation for this phenomenon. The answer would be, the addition of substrate causes more EX complex, which leads to more intramolecular forces in the transition state and more stability. On to the next question. Why is there a high degree of stereospecificity in the interaction of enzymes with their substrate? This was introduced in topic 48 in chapter 9.1. It's referring to the active site, and some key terms to think about would be catalytic residues within the active site and interactions between the substrate and the enzyme? The answer is side chains in the active site need a complementary fit between the enzyme and the substrate to form intramolecular forces in order to catalyze the reaction. An example would be in chymotrypsin, where you can see the active site has a deep pocket and no polar charges. So what amino acids would have a complementary fit with the side chains in this active site? The answer would be nonpolar and large amino acids. For an example, tryptophan, tyrosine, phenylalanine, and leucine. Another example is trypsin. You can see this also has a deep pocket like chymotrypsin, except the difference here is it has a negative charge, which is polar. So what amino acids would complement the side chains in trypsin? The answer would be amino acids with a positive charge that are large: leucine and arginine. And for the last example, you can see elastase has a very shallow specificity pocket with a positive charge. So what amino acids would fit best in this pocket? That would be alanine, valine, and glycine. So that concludes my review. Good luck on your exam, everyone.